Dear colleagues, first of all, I would like to thank the organizers for inviting me to participate in this exciting congress. And it's my great pleasure now to review with you what's new in genetics. Let's look at these three gentlemen who all have Parkinson's disease and what's special about them. So they have very different histories. As you can see, they're all about the same age. Ages of onset, however, vary a lot between 13 and 55 years, as do the levodopa equivalent doses. They all carry mutations or pathogenic variants in genes associated or linked to Parkinson's disease, as you can see here, Parkin, GBA, and LRRK2. And what is new, really, is that for at least the GBA and the LRRK2 mutation carriers, there are now gene-specific clinical trial in progress. In the next few minutes, I will report on new insights in genetics, review some new and old questions, and then finish off with new translational perspectives. Let's start with the new insights. Very recently, a large, probably the largest so far screening study for mutations in Parkinson's disease genes has been completed. And so far, 11,000 patients have been screened at about 100 centers. And the outcome is that 8.5% of the patients, PD patients, carry variants, pathogenic variants in the GBA gene, 2% in the LRRK2 gene, and 0.0% in the Parkin gene. And the first results of this study have already been published. Further new insights come from the MDS gene database. So this database brings together geneticists, clinicians, epidemiologists, students, bioinformaticians, and technical staff from more than 100 members, with more than 100 members in more than 20 countries. And this is a project that is sponsored by the International Parkinson's and Movement Disorder Society. And the aim of this big project is to collect all of the published information on clinical and genetic features of Parkinson's disease available in the English literature, with specific information on genetics and on the clinical features of an individual patient. And then you can go and use this not just as a database, but also to filter your material that you're interested in, and also to obtain some summary statistics. And this is what you can see here about the clinical part of the database. So you can see it's a little bit small, but you can go online and check this out. And then you'll see that you can look, for example, for different clinical features or ages of onset or distributions of mutations, etc. And then there's also more detailed information available on the mutations that you can see and find in the mutation part. All of these data have also been published in 3 different reviews, one on the dominant forms, one on the recessive forms, and one just very recently in Press Now reviewing the atypical Parkinsonism genes. Another large project that I would like to mention here is the Michael J. Fox Foundation Global Genetic PD Cohort Project. And this includes 4,000 mutation carriers, for each of whom detailed clinical and also genetic information is available. And more than 90 centers from all inhabited continents are taking part in this large study. All of the clinical information has been carefully curated, and pathogenicity scoring has been performed of all variants. And again, all of this material has been put and entered into a searchable database, which will soon be publicly available. So let's now turn to the second part and review new and old questions with respect to Parkinson's genetics. So first of all, how about ethnicities, origins, and ethnicities? And you can see here, and this is data stemming from this Michael J. Fox project that I've just shown to you. And this is just an overview on the different countries included. So you can see a lot of different countries involved in this large study, and you saw this on the world map before as well. However, when it comes to ethnicities, we still have information mostly available on white Europeans. And you can see this here. So by far the largest piece of this pie, 2/3 actually, are whites. And so this is, of course, a problem because we don't really know a lot about other ethnicities. And it is very possible that we might find very different genetic frequencies, and perhaps also very different clinical presentations. Another question, which is also a bit old, but still new and still relevant, is the quality of the genetic testing. And you can see here we've put together how these different colleagues in all these different countries and centers have assessed their patients. And you can see the blue dots, they stand for candidate gene sequencing. And you see here the 6 most frequently tested genes on the x-axis, and you can see that really by far the majority of mutations have actually been found still by candidate gene sequencing, and panels, PD panels, these would be the red squares, are still relatively rare. If you see here, they hover around 20%. Another important new and old question is the variable expressivity of the clinical presentation. And I would like to show this to you here with an example. So both of these patients carry a homozygous PINK1 mutation. And you can see here the first patient is a German patient with a late age of onset in her early 60s. And there's actually nothing atypical about her Parkinson's. Whereas the little boy here, and the video was kindly shared by colleagues from Saudi Arabia, he has a very different clinical picture and an age of onset of only 5 years, also carrying a homozygous PINK1 mutation. He does respond to treatment, as you can see here, but it's a very, very different clinical picture, as you will agree, I'm sure, and he also has quite prominent dyskinesias. Another important question, and still mostly unresolved, is that of reduced penetrance. And for this, I would like to share with you some of our own unpublished data. And you can see here in this pie chart in the blue part, we have screened Parkinson's disease patients for Parkinson's disease mutations. Very straightforward. And you can see, yes, this dark blue part here of this ring shows you how many PD mutations we have found in PD genes. Interestingly, in the red pie part, we have also found Parkinson's disease mutations in the dystonia patients that we included. And perhaps most interestingly, when you go to the gray part, you will see that even among the controls, and these were more than 4,500, you can see that even here, in a considerable proportion of elderly controls, we also find Parkinson's disease gene mutations. And we know very well that penetrance is reduced, especially in the dominantly inherited forms of Parkinson's disease. Another important old new question is that about genetic modifiers. And you will perhaps be familiar already with this very, very elegant and comprehensive study that was published in the end of 2019. And that's put together 90 independent risk loci. And not only that, but it also performed a subgrouping of these patients who then had different sets of these risk loci and grouped them into quartiles. And you can see that patients that fall into the second risk quartile, so they carry more of those risk factor genes, that they have that their risk to develop Parkinson's disease is about doubled. Whereas those that are in the third quartile already have a risk that is about 4 times as high as those without these particular risk SNPs. And then when you look in the highest risk quartile, the fourth quartile, the risk goes up to about 7 times, 7-fold. And so this is very interesting, but still something that we need to understand a lot better, and which could also potentially be applied for stratifying patients for clinical trials or research studies. Let's now finish off with the new translational perspectives. First of all, I would like to mention the clinical trials that are ongoing, and I alluded to this already at the very beginning of my talk. And you can see here from a very recent paper by Mullen and colleagues who looked at the response of patients with GBA mutations and those without, all Parkinson's patients, who received Ambroxol. Ambroxol, a cough suppressant, as you probably know, and which increases levels of GCase, of the protein encoded by GBA. And indeed, they were able to show this. And they were now here comparing baseline and data at about 6 months. And you can see that indeed, the GCase protein levels do go up, as was hoped for. However, what is also interesting, that there was no difference between the carriers of GBA variants, pathogenic variants, and those who did not have pathogenic GBA variants. And so this would allow for 2 potential conclusions. So either this is just simply non-specific, or it doesn't matter whether you have a mutation or not. And from this would result a second potential conclusion, which would be a very nice one, in fact, and that could mean that also patients without pathogenic variants in these genes might benefit from this particular treatment. It's a small, uncontrolled study, though, which will have to be repeated and is ongoing in a larger and more controlled setting at the moment. Another study that I would like to mention is one that we are currently conducting here at the University of Lübeck. And again, we're using genetic stratification for our clinical trial. And as you can see here, we have grouped them into a mito plus versus a mito minus group. And this refers to mitochondrial disturbance, mitochondrial genetic risk load. And we are starting off with those who are homozygous or compound heterozygous for mutations in the mitochondria-associated genes Parkin or PINK1 and have 2 mutations, so biallelic mutations plus. Then the next group would be those that carry just a single mutation, heterozygous carriers in one of these 2 genes, followed by a group where we applied such a polygenic risk score as I showed you in the modifier slide, and this would be our mitochondrial omics group, and those we compare to those that have neither. And this is now ongoing and has been published, the protocol, and we're using coenzyme Q10 in these patients, and it would be very interesting to see whether these different groups respond differently, and it's a placebo-controlled trial. Finally, I would like to point out that there are really large networks now. I've alluded to some, the MDS gene at the beginning, and also the Michael J. Fox initiative, but there are others. There's PD Generation that I would like to mention, which is led by the Parkinson's Foundation. There's the GOPD initiative. And then most recently, there's the GP2 program, which stands for Global Parkinson's Genetics Program, which also is aiming at understanding the genetic architecture of Parkinson's disease and is going to genotype 150,000 patients. With this, I would like to sum up. There are now very large databases. This is new to that extent. Largest genetic screening studies and networks that we ever had. It's important, though, to see that we're still looking at select ethnicities, that there's still limited genetic testing, variable expressivity, and reduced penetrance that we need to understand better. There are first clinical trials and very large global networks that we can all engage in and all work together. And with this, I would like to finish off and thank you very much for your attention, and also thank the team at home for all their excellent work. Thank you very much.